My name is Craig Haslow. I'm the Chief Scientific Officer at CellCan, Regenerative Medicine and Cell Therapy Network. And on behalf of the Clinical Translation Education Group, I'd like to welcome everyone today to our workshop. Thank you so much for traveling afar, or even if it was not much of a journey for you. We're very pleased that you join us. This year, our edition is Current Trends in Biotherapeutics Workshop. Really, the state of affairs have changed dramatically in our field, as we're all aware, and the ecosystem has evolved so rapidly within even the last 2 years since our first offer. It's amazing to see the progress that's been made. Now in Canada, we have these 3 products that are market authorized in the cell and gene therapy sector, which is extremely exciting, and we expect to see many more in the very near future. A little bit of a history of the Clinical Translation Education Group. We came together somewhere before 2017 as a group of networks, federally funded or provincially funded, all with commonalities around promoting cell and gene therapies in the regenerative medicine, cancer immunotherapy sectors, and realized that our training opportunities and the offers we're putting together had so much synergy that it only made sense to put forth a united front on common workshops. Initially, we had the bench to bedside for biotherapeutic scientists in 2017. Last year, we had our IP for entrepreneurs, and this year, we're moving into a scan of what's here and now in biotherapeutics and what's on the immediate horizon. We're very excited about the program that's been put together today. We have an excellent lineup of speakers, and I think you'll all agree it will be a tremendous workshop. Now, there's a few housekeeping items that I need to mention as well. We encourage, this is absolutely mandatory. We encourage interaction. Please talk to one another, get to know each other. Don't hesitate to speak to any one of our speakers when you have a chance to. I particularly encourage meet with people that are a little bit outside of your background and expertise, and get to know one another, because the more we talk, the more we realize it's a bit more of a close-knit and small community. The flow of the workshop for the day will be broken up into our morning sessions, starting with our How Technology is Changing Preclinical Research. It'll move into our Trends in Manufacturing after the break at 10:30. We'll have a lunch buffet, which fortunately, if you're a new parent like myself in the crowd, you're happy to eat a little bit early. And we'll roll that right into our Keynote lunch session. After the lunch, there'll be a Future Trends in Translational Medicine section with 2 excellent talks, followed by a networking break, and we'll get into an If You Build It, They Will Come session in the afternoon, followed by a Meet the Experts panel session. So we're very excited. I'd very much like to point your attention to the hashtag in the bottom left corner. Please feel free throughout the morning to use social media. Use the hashtag CTEG 2019, CTEG 2019. We have a list of Twitter handles and social media handles that are readily accessible in the program. By all means, reach out to everyone, and we certainly want your feedback. So at the end of the session, there'll be a survey circulated. It's absolutely critical for us to develop our metrics and understanding of what works in our workshops, what types of areas you'd like to see in the future, and it makes a lot of our organizations very happy. So if you could take the time to fill that out, we'll collect those by the end of the day. I have to send a special thanks to our sponsors. Without them, these events are very difficult to put together. In particular, the slide just scrolled. Our gold level sponsors, the Center for Commercialization of Cancer Immunotherapy and Regenerative Medicine, C3I of Montreal, GE Healthcare, the University of Toronto, and Medicine by Design. Thank you all so much. We really appreciate you being part of this. So you'll see our members of our CTEG group. It's the original 6. It's certainly not an ultra-exclusive group. We welcome new partnerships at any time. But the core comprises of BioCanRx, Canada's Immunotherapy Network, CCRM here in Toronto, the Center for Commercialization of Regenerative Medicine, our organizations, CellCan, Regenerative Medicine and Cell Therapy Network, Ontario Institute for Cancer Research, the Ontario Institute for Regenerative Medicine, and of course the Stem Cell Network. So thank you, everyone. Please don't hesitate, like I mentioned, to come up, have a chat, introduce yourselves, and we'd really like to get to know each other.
Thank you for the opportunity to speak for a couple of minutes this morning, and for having us as a sponsor this year. Just putting up our confidentiality and disclaimers. The promise of cell therapy. So this is a cell therapy manufacturing workflow. Okay, so this is a cell therapy manufacturing workflow as GE Healthcare envisions it. I'd like to point out that we have various products, and throughout this workflow, most of our products you can see are multifunctional, and you can see them throughout this workflow. The first one I'd like to point out is the VIA freezer. This is a liquid nitrogen-free controlled rate freezer, and you can see that it's used in the apheresis collection at the patient sites, where you can freeze down your patient product. And you can also see that towards the end of the workflow in cryopreservation, once your product is made, it can be frozen down in this freezer. The other thing I'd like to point out in the next step is the isolation step. Here you'll see that we use Ficol-Pac, which is one of our density gradient medias, and it's used with centrifugation to isolate cells such as your T cells or your mononuclear cells using our CPAX instrument. The CPAX has various protocols, and it allows you to use it throughout the workflow. So in isolation, you can also do bead addition, washes of your cells. In the downstream, you'll see it's used to harvest your cells after you've expanded your genetically modified cells. And also it's used to prepare your final formulation of your product and put into bags. The other thing I'd like to point out is the Zuri cell expansion system. This is a rocking bed platform bioreactor. It has a cell bag on it. It's a closed system. This is where you would expand your genetically modified cells. We also offer interleukins, specifically interleukin 2, 15, and 21, and we sell a specific T-cell expansion media. As you can see, all of these are closed systems and can be applied in clinical settings. Because every cell matters. Thank you.
Thank you very much, Barbara. And now I'll turn it over to Allison Brown. She's the Director of Strategy and Translation at Medicine by Design.
Hi, good morning, everyone. Thanks for the introduction and for the opportunity to say a few remarks this morning. I joined Medicine by Design in April of this year, so it's been a busy few months since joining the team, and I wanted to take the opportunity just to tell you a little bit about our plans moving forward. As most of you know, Medicine by Design was launched in 2015 with a $114 million grant from the Canada First Research Excellence Fund. This was the largest grant ever given to a Canadian university, and since 2015, we have funded 128 investigators at the University of Toronto and our affiliated hospitals. We've funded over 400 trainees and launched over 100 projects focused on regenerative medicine, which includes the cell and gene therapy space, as well as strategies, both small molecule strategies and biologic strategies to enhance endogenous repair, and also investing in next generation enabling technologies for the regenerative medicine space. Our first round of large multidisciplinary, multi-institutional team projects were launched in 2016. 19 projects were funded to the tune of $27 million. And those projects have just come to a close. And as they've started to close out, we will be in the next few days announcing our next round of strategic investment into 12 projects, which primarily build upon the successes of our Cycle 1 Team Project Awards. And really between now and 2022, which is when our first round of funding from the federal government ends, we're focused on sharpening the peaks of excellence within the Medicine by Design portfolio. The projects that we will be funding build upon Cycle 1 successes. We still are investing in the cell and gene therapy space and endogenous repair and new technologies, and really focusing on how enabling technologies can help us ask important biological questions in the regenerative medicine space to ultimately inform new therapeutic strategies. An important component to this next phase of team initiatives is a focus on translating our research outcomes into impact. And with that, focusing on how we can resource these teams around translation. And even beyond the broad spectrum of translation and commercialization, we're starting to think about and talk with these teams about how we can support researchers at the earliest stages of translation. So far beyond thinking about that phase 1 clinical trial, how can we, as researchers and as trainees, start to think about product ideation and unmet clinical needs very early on in the research process. So we're doing that in collaboration with our commercialization partner, CCRM. And we've also started to forge additional partnerships in the Toronto ecosystem, including TAP, which is formerly Mars Innovation. We're engaging with the investment community and really with a whole host of product development specialists, not only in regenerative medicine, but also in adjacent fields that can bring expertise and act as mentors for these teams moving forward over the next 3 years. For the trainees in the room, and some of you I know are involved in Medicine by Design projects, we're also bolstering our partnership with entities like the Creative Destruction Lab, as well as some other commercialization engines within Toronto to provide unique opportunities for students to start new companies, to gain mentorship around how to commercialize ideas. So you'll be hearing some new news around those concepts soon. And with that, I just wanted to say that I'm delighted to be here today and looking forward to the program.